Hi everyone, we are going to talk about glycogen metabolism in this chapter. Glycogen is a readily mobilized storage form of glucose. It is a very large branched polymer of glucose residues that can be broken down to yield glucose molecules when energy is needed. Signaling catalysis leads to the mobilization of glycogen to produce glucose, an energy source for cyclists. So let's talk about the glycogen. And the dark particles in the cytoplasm shows here are glycogen granules. At the core of the glycogen molecule is the protein glycogen, shown in yellow. So each line represents glucose molecules joined by an alpha-1,4 glycosidic linkage. The non-reducing ends of the glycogen molecule form the surface of the glycogen granule. Degradation takes place at this surface. In this structure of two outer branches of glycogen molecule, the residues at the non-reducing ends are shown in red here, and the residue that starts a branch is shown in green. The rest of the glycogen molecule is represented by R. So let's see the fate of glucose 6-phosphate. It derived from glycogen, and it can be used as a fuel for anaerobic or aerobic metabolism, as in, for instance, in muscle. It can also be converted into free glucose in the liver and subsequently released into the blood. And then it can also be processed by the pentose phosphate pathway to generate NADPH or ribose in a variety of tissues. So let's take a look at the glycogen breakdown, and it requires the interplay of several enzymes. Phosphorylase catalyzes the phosphorylated cleavage of glycogen to release glucose 1-phosphate. Glycogen phosphorylase is the key enzyme in glycogen breakdown, cleaves its substrate by the addition of orthophosphate to yield glucose 1-phosphate. The cleavage of a bond by the addition of orthophosphate is referred to as phosphorylases. So mechanism is a paradox phosphate participates in the phosphorylated cleavage of glycogen. A bond hydrogen phosphate group, shows in red here, favors the cleavage of the glycosidic bond by donating a proton to the carbon-4 oxygen of the departing glycosyl group, shows in black here. This reaction results in the formation of a carbonium ion and is favored by the transfer of a proton from the protonated phosphate group of the bond pyridoxal phosphate, short for PLP group, and it shows in blue here. The carbonium ion and the orthophosphate combine to form glucose 1-phosphate. A debranching enzyme also is needed for the breakdown of glycogen. First, alpha-1,4 glycosidic bonds on each branch are cleaved by phosphorylases, leaving 4 residues among each branch. The transferase shifts a block of 3 glucose residues from one outer branch to the other. In this reaction, the alpha-1,4 glycosidic link between the blue and the green residues is broken down and a new alpha-1 4 link between the blue and the yellow residue is formed. The green residue is then removed by alpha-1 6 glucosidase, leaving a linear chain with all alpha-1 4 linkage, suitable for further cleavage by phosphorylase. Phosphoglucomanase converts glucose 1 phosphate into glucose 6 phosphate. The catalytic site of an active mutase molecule contains a phosphorylated serine residue. The phosphyl group is transferred from the serine residue to the carbon 6 hydroxy group of glucose 1 phosphate to form glucose 1 6 biophosphate. The carbon 1 phosphorus group of this intermediate is then shuttled to the same serine residue, resulting in the formation of glucose 6 phosphate and the regeneration of the phosphoenzyme. As phosphorus group is transferred from the enzyme to the substrate, and a different phosphorus group is transferred back to restore the enzyme to its initial state. The liver contains glucose 6 phosphate, a hydrolytic enzyme absent from muscle. The liver contains a hydrolytic enzyme, glucose 6 phosphatase, then enables glucose to leave that organ. This enzyme cleaves the phosphorus group to form free glucose, or orthophosphate. The glucose 6 phosphate is the same enzyme that releases free glucose at the conclusion of glucogenesis. Phosphorylase is regulated by allosteric interactions and reversible phosphorylation. We will examine the differences in the control of two isogamic forms of glycogen phosphorylase, one specific for liver and one specific for skeletal muscle. Both phosphorylase B and phosphorylase A exist as equilibrium between an active R state and a less active T state. Phosphorylase B is usually inactive because the equilibrium favors the T state. Phosphorylase A is usually active because the equilibrium favors the R state. Regulatory structures are shown in blue and green here. The binding of glucose to phosphorylase A shifts the equilibrium to the T state and inactivates the enzyme. Thus, glycogen is not mobilized when glucose is already abundant. A low energy charge, represented by high concentration of AMP, favors the transition to the R state. ATP and glucose 6 phosphate stabilize the T state. Okay, so that's all for today, and thank you for your listening.